I'm endlessly fascinated and curious about the world around us, about business, about politics, about the arts, about culture, developments, social issues, and the people. Most importantly, about the people that are shaping our future. Every night, I get the opportunity to have conversations, to dive into conversations with thought leaders, with change makers, entrepreneurs, politicians, and everyday heroes who unpack the ideas and stories that I think matter to all of us. What's the future of our economy? What's the future of our country? How do we build stronger communities? What can we learn from music, theater, sports, politics, the arts? How does policy shape our real lives? What does leadership look like, real leadership in today's environment? If it's interesting and relevant and topical and we're talking about it, you'll hear about it here at 6 o'clock on the Brian Crombie Radio Hour. I want to have real conversations, in-depth conversations. These are ideas we're sharing. This is the Brian Crombie Radio Hour. Thanks for joining me.
Good evening, everyone, and welcome to the Brian Crombie Radio Hour. I've got a really interesting gentleman, company, and at least two fascinating, innovative pharmaceutical medical products to introduce you to tonight. Murat Croci is with us tonight. He is the co-founder and co-CEO of Ibex Biosciences. It's an innovative biotechnology company. And what they're all about is they want to discover, validate, and produce potential solutions to a multitude of serious challenges facing human health by investing in exploratory projects and processes that may not have been validated with the public research community. He's just told me a little bit out, and I think he's going to tell you about two products that he's taken through the process, and now he's struggling to find the money to commercialize them. Murat, welcome to the show, sir.
Thank you, Brian. Welcome to be here. Great to be here.
So tell me about these two products that you've done a lot of work with, if you could.
Certainly. As you said, the company is trying to go after stuff that's very difficult to cure. So we decided to go after malaria, but unlike other drugs in the market, we found an Achilles heel. So you have these parasites, and they basically, I won't go into the details, and maybe your listeners would care, but I'll do it in 30 seconds. Let's say you're bitten by an infected mosquito. That mosquito goes from your blood into your liver. It hangs out there for about 10 days, and it comes out of the liver. Now, you get no symptoms from this. It's asymptomatic. But once it goes in the bloodstream, that's when the symptoms occur. That's when they start attacking red blood cells. So what they do is they enter red blood cells, reproduce, red blood cells explode, and they infect new red blood cells. And this goes on and on and on until either your immune system clears infection or you have organ failure and you die. So our innovative solution is extremely simple. We found out how to plug the hole so they can't get into a red blood cell. Well, guess what? They have 30 seconds to a minute to get into a red blood cell. If they don't do that, they perish and the infection ends. And that's simple, and it solves the problem, both prophylactically and it also serves a purpose to cure. This has been tested in mice with human blood. So it replicates the human state. So that's how we've solved malaria. So that's what we're doing. We're trying to seek funds to get this through a human trial. Human trial would be very simple. You infect the patient and you administer the drug or vice versa, and you see if it works. In 3 days, if it clears infection, then it works, and you basically solved one of the world's most difficult problems in medical humanity, which is malaria. It kills 600 people every year. It's killed millions throughout history, if not billions.
So tell us a little bit about malaria, just to take a step back, if you could. It's carried by mosquitoes?
Yeah, it's a parasite. It's called Plasmodium falciparum, and it doesn't infect all mosquitoes. So people say, oh, you can get bitten by a mosquito, right? You can get malaria, right? That's not the case. The mosquito would have to be infected with the parasite in order to get malaria. And it's pretty much non-existent in the US because it's been eradicated. You might hear one or two cases here and there. Most cases are people who come back from Africa and are infected because it won't show up until like 10 days. In fact, there's a Fox News host.
Yeah, how has it been eradicated in North America versus not in Africa?
Well, it was here in the US, in America, like a long time ago, right?
Yeah, but how has it been eradicated? What was the process?
I mean, through through excessive uses of pesticides, which have killed everything. Plus, we don't have wet seasons like they do in Africa. So the climate's a little different. It's warmer there, it's wetter there.
Okay, so still a big problem in Africa, obviously.
Right. Africa, Indonesia, some places in the Middle East, South America. So yeah, you have fatalities and not only death, you have loss of work hours. You have to take care of family members. You have to stay home for weeks. You have massive headaches, fevers, shivers. The symptoms can be severe. And you take a drug and you're also going to get you can get side effects from the current drugs that are available. So it's not a pretty sight.
There's no vaccine for malaria. Is that correct?
So there are vaccines for malaria, and they're about 70% effective, and you need 4 shots. And they're seasonal. A parasite can evade vaccines more so than other types of infections. That's their hallmark. They can do that. But the current vaccines are woefully inadequate. Even the ones that have come out, they're, again, 70% effective, 4 injections. So
And if you're infected and you get sick, what do you take then?
like artemisinin is a standard of care right now that people use for severe malaria. That may or may not work. It's not 100% effective. It has side effects. It's inadequate. And the mosquitoes have developed resistance to that drug right now. So now they're trying to find another drug to replace it because it's not as effective as it was like 10 years ago.
So it sounds like your invention, your product is world changing.
Yeah, I mean, they can't evolve. It's impossible. You have to exist to evolve. They can't get into a red blood cell, they die. So it's pretty straightforward. It's extremely simple. It's a simple solution to this huge problem. Maybe Bill Gates is listening here and says, I want to go and invest in Ibex and solve this problem. I mean, it's just sitting there. And I say these things with certainty because if your listeners happen to go to the site, they can actually see these reports that were generated. I actually published them that were done by third parties. And they're research organizations that actually work with Bill Gates and Gates Foundation's partners, like MMV, Medicines for Malaria Ventures. They're the ones, independent lab that did these tests, that has validated in the actual report, cured, one-shot cure.
How'd you come up with this technology? Is this your invention? Is this from an academic institution? Is this how'd you come up with this idea to stop them from being able to go into the red blood cells?
Great idea. A great question, Brian. Brian, look, when we started the company, we wanted to do things very differently than everyone else. So we actually commissioned these experiments from scratch. We didn't license these from anywhere else. We didn't license it from another university or institution. I went and I basically found scientists who would take risks and chances and who are innovative. Our chief scientific officer, Dr. Vidal Dela Cruz, invented the phage display technology, a technology that won the Nobel Prize. I have Dr. Norman Lai, who worked for NIH under Roscoe Brady. His lab generated products that generated in the billions of dollars when they were licensed to large pharma. So I sought these scientists. I asked them to conceptualize ideas to solve major problems. And we came up with this solution for malaria. And we came up with another solution for cancer. And I guess we'll talk about that a little later.
Yeah, let's leave that for just a second. And so you've been doing this, you said, for 8 years?
10 years.
10 years. And how much money have you raised so far?
So we've raised between equity investments and loans, it's close to $20 million.
And how much are you trying to raise right now? Because this would be a substantial change to take it out of animal testing into human testing, would it not?
Absolutely huge. So we were trying to raise in total 25 million, and that would cover colon cancer, refractory colon cancer, and malaria in a phase 1 study. So we're doing 2 indications. We're trying to raise 25 million in a Reg D 506C offering. And so the malaria would be very quick because it's an acute indication. Cancer would take a little longer time. But our drug, and I don't want to jump the gun here. It's probably jumped a lot of guns here, Brian. But I mean, look, the cancer, we'd probably go after patients who are very sick, maybe who are they're fit, but they're basically on their last leg in terms of trying other new drugs that are out there, and so that we'd go after those sick patients in that clinical trial. So we'd hope to get that done as quickly as possible as well. So
let's stay with malaria just for another minute, if I could. Please explain to everyone, if you could, what the process to commercialization would be. You're talking about funding the phase 1. Would you need then to do a phase 2 and a phase 3 before you can submit to the FDA to get approval? And how many people would be in your phase 1? What would the phase 2 do, and how many people? And what would the phase 3 do, and how many people? And how long would that take before you would likely get approved by the FDA? Give us the roadmap, if you could.
Great questions. Great question. There are 3 places in the world that you can do a challenge model, and one of them is in our backyard at NI, National Institutes of Health, and they will run a challenge model. That means they actually raise Plasmodium falciparum. They culture it, and basically through a CRO, a contract research organization who's worked with them in the past and have worked with many phase 1 clinical trials dealing just with malaria, we would contract them, and they would seek volunteers, which they tell me is not very difficult for malaria, believe it or not. And these individuals will get our drug administered and will get live malaria injected into them, so they will get infected. And at which point, within 2 or 3 days, it will be determined whether the drug works or not. And then, of course, there's the filings that go along with the results that would be submitted to the FDA. And if it cures these patients like we think the drug will, we essentially pass phase 1 studies.
And so phase 1 is efficacy in a small population. Is that correct?
Correct. I would say 20 people. 20
Excellent. Okay.
people.
So then what's phase 2?
Would be a field test, Brian. That we would need to go to Africa or another place in an endemic region, and we haven't done the study design there, but basically it would at this point we would raise additional funds, so that would be easy. If we pass a phase 1, we can get institutional funds from almost anywhere, so that wouldn't be a problem. But
okay, and then a fairly short, I know you haven't done this, the study design, you say, but is that a large population and a long time, or is that, again, a small population, a short time?
Yes. So there's no shortage of those who are infected if you go to Africa.
Right.
I couldn't tell you the exact timeline, but I would say that probably it's going to be not 20, but maybe 100 patients that would be injected with our drug, Ibex 13, to see if it works with them. And we would monitor those patients. And again, the drug is supposed to clear the infection in 2 to 3 days. So the clinical trial shouldn't take a long time.
Right. Okay. And then the phase 3, I've always been told the phase 3 are the big, long, big population, expensive ones. Would you probably have that situation as well?
I mean, it depends on the indication, right? This is an acute indication, so you have the infection, and after the drug is administered, you shouldn't have it. So you're talking per patient, maybe 3-4 days, and then you'll monitor them several weeks afterwards. So I wouldn't think that would be very expensive either. But at that point, it's kind of moot because they know the drug works.
Yeah,
and you have no shortage of investors. But in terms of timeline, I would probably say that would probably take about a year to do a phase 3 from start to finish. And most of it's paperwork, I would say. It's not actually administering the drug to patients and monitoring.
So you're talking about, what, 3 years to 5 years before this product would come to the market?
Yeah, I'd say like probably 3 to 5 is correct. That's a great range.
Okay. And how many people did you say perish every year from malaria?
It's about 600,000.
600,000.
Yeah, a
And
year.
and everything else you say is either 70% effective or doesn't actually solve the problem. And what you're looking
That's right.
for is $25 million to do the phase one that starts the process, gets the proof of efficacy, and then after that you got to prove that it's safe. You got to do it in Africa, and you got to do the phase three, and you got to submit it to the FDA and get approval. So there's processes to go through, but the real nut is getting this $25 million to do the phase one to prove it's efficacious. And you've proven already that it's efficacious with human blood, albeit in mice. Is that correct?
That's absolutely right. And just to clarify, it's 5 million just for malaria. The rest, the rest is for the cancer program. So there's two
$5 million.
indications.
And effectively what this is an option on solving problems for 600,000 people a year.
Yeah, I mean, that's what
Unbelievable.
I'm saying. I mean, if like Bill Gates is spending much more than this to resolve this, and if he wants it solved, here we go. He can do it immediately. I mean, anyway, it doesn't have to be Bill Gates. You know, Elon Musk got malaria. He almost died from it. Actually, it's well reported in South Africa. Could be any one of these guys. They want to solve this problem. This is the way to do it. It's a definitive solution to malaria. They cannot circumvent, the blockade. And we happen to find the drug that pinpoints that exact point where it can't enter. They just perish. So it's remarkable that we discovered it and validated it like we have. We're going to file a publication next year. We're submitting a manuscript for this approximately. It takes a while to publish, but all the data is available online in our website. So yeah.
Fascinating. We're going to take a break for some messages and be back in just 2 minutes. And I'm going to ask Pierre-Jean, the banker, to talk a little bit about the challenge of raising money and what he thinks of this product and this company. Stay with us, everyone. Back in just 2 minutes. Welcome back, everyone, to the Brian Crombie Radio Hour. I've got Murat Croci from Ibex. We've been just chatting about his product for malaria that sounds actually fascinating. And I've got Pierre-Jean. I always mispronounce your last name. I apologize. Fournier, I think it is, who is the investment banker that's taking Murat and his company to the market. We've had the pleasure of chatting with Pierre-Jean, PJ for short, before. He runs an investment bank out of Paris, France, but he's got operations in London, England, and in the United States, and he raises money for people around the world that are fascinating companies. Pierre-Jean, why is Murat having challenges raising $25 million? With you on the assignment, it should be easy. No, seriously, tell us how challenging it is for biotech companies to raise money.
I will answer to it, but in 1 minute, I want to tell you why is it that I took the challenge to work with Murat and Ibex.
Please.
Murat is a very intelligent individual, for sure. He has no, if any, scientific background. Still, if you follow what he said during the first half of the presentation, it was very easy to follow. He takes the right verbs for you to understand. So I took the challenge of fundraising for Murat because he's a good individual. Also, he wants to change the world, and that's what he's about to do. Not only malaria, but also in cancer, colorectal cancer. So it's challenging because the time are very difficult. We published a research in my bank about 2 weeks ago, and we demonstrate that it takes about 22 months to raise funds in the biotech world. That is the current time now. So no surprise, we made some time. Now we got traction on the cancer part of the business, but it is expensive. There's a lot of money to put on the table. We have to raise $20 million, but we have some strong mark of interest. Besides, is working in a segment of biotech which is called ADC. ADC, well, my friend, I have cancer, and when I was first treated with cancer, I did chemotherapy. Chemotherapy is like bleach that goes into your blood and kill all the cells that are moving too fast, like your hair, like your cancer cells. And in this case, this technology select the cancer cells and try to destroy the cancer cells very selectively and effectively. That's why I was very interested into the project.
Murat, why don't you tell us a little bit about this cancer product then, if you could?
Okay, an antibody drug conjugate is an antibody with a linker. At the end of the linker, there's a drug, like a chemo agent. And the antibody's objective is to bind to the cell surface of the tumor and then enter the tumor. And when it enters a tumor, they call that endocytosis. That's the process where the antibody drug conjugate enters the tumor cell surface. And then once it's inside, it releases its payload. Enzymes clip the payload. So the payload's a chemo agent, and the chemo agent disperses inside the tumor and destroys it. So that's what an ADC is. Now, this is a relatively new area of therapeutics, and they've shown to be very effective. Most recently, they talked a lot about Enhertu for low HER2 breast cancer, which is an ADC. And so you're going to see a lot of these drugs that are going to come out into the market, but they're not all the same. Some of these drugs achieve something called bystander effect. So not all of the conjugate gets inside the tumor. Some are released early on, and the chemo agent that is near the tumor happens to kill the tumor cell. They call that a bystander effect. In our case, we're working with alpaca-based antibodies. They're called VHH, so they're considerably smaller than a traditional heavy light chain. And so that provides a benefit for targeting small crevices on proteins that may be difficult to bind to. And then there's another advantage that our drug has, okay? And that is endocytosis. That means it really goes into tumor cells much readily than other proteins. So it's called CD147. You don't have to remember that. But because it's targeting that CD147 protein, it happens to go inside cancer cells very, very, very effectively. When I mean effectively, I mean how effectively? Well, we've done, the most recent study was they grafted a human tumor into a mouse. They call these xenograft models. And then they administered the ADC intravenously into the mouse. And then they monitor the size of the tumor, because these tumors will get very, very large. They have no immune systems in these mice, so it's continuing to grow. Well, in the administration of the Ibex 13 ADC, there was no tumor left. It didn't just suppress tumor growth, it eliminated the tumor. They couldn't find anything there. It was 100%. And just like the malaria study, I upload these reports publicly on our website through the portal. And so, because it's very difficult to believe, it's almost incredulous when you say something to someone and he says, well, here's the independent data. This is what this drug achieved. I didn't say it. The independent party says it. And what's even more interesting here is that the drug, the cancer type that we tested, was highly chemoresistant to all the 5-fluorouracil and oxaliplatin. These are chemo drugs that are standard of care drugs given to colon cancer patients. And when it becomes refractory, so those drugs work, okay? Don't get me wrong, right? 65% of those patients that receive it go into complete remission. That means 5 years it doesn't come back. But when it comes back, those cells become resistant. And then the 35% struggle to get better, and they go on different regimens, and the cells keep on coming back. That was the cells that were implanted in this animal model, those chemo-resistant cells. And it took the tumor into zero. So we are going into refractory colon cancer. And as I said earlier, which I jumped in, we want to target patients that are terminal, that because they express this target more than patients who could receive chemo.
Fascinating. So let me ask you a couple of questions if I could. So it sounds like you're not actually changing what you call the payload. The payload is still chemo. You've got what? Something that is the carrier of the payload that can get into the cell, into the tumor more effectively. Is that correct?
That's correct.
So it's a technology that you've got rather than a new drug.
Well, the antibody is a carrier, and you're using a standard payload, which is correct. But together, they called an ADC, an antibody drug conjugate. So the type of drug we're using, Brian, will not work unless it enters the cell. If it doesn't enter this cell, it won't work. So the carrier is extremely important.
So
That
getting
endocytosis.
it into the cell is the key. And so tell me, why are so many cells chemo-resistant?
Well, one of the mechanisms is exactly why our drug works so well, because it overexpresses CD147. The literature is huge on this. This is a mechanism by which the cancer cells resist traditional chemo agents. And we're exploiting that. We're noticing that. We looked at the publications and cells that are chemo resistant, and we determined, like, what are these cells overexpressing to defend themselves against chemo agents? And we noticed the expression levels of 147 on the cell surface was tremendously high, particularly in chemo-resistant colon cancer. And so that's what we targeted, and it works wonderfully. I mean,
And so you're
it's like,
focused right now on colon cancer, refractory colon cancer to be exact, I think you said. Is this technology and this drug combination effective in other cancers as well, or we don't know yet?
yes, it would be effective in other cancers. We tested in melanoma, we tested in ovarian cancer, and they were both effective. But we just have to choose one indication to begin with and then move from there.
And did I understand what you said a couple minutes ago that 35% of the people, so chemo works in 65% of the people, but not in 35% of the people, and in 35% of the people, it comes back within 5 years? Is that
That's
what
right.
you said?
That's right. That's right.
That's
right.
incredible. I've never heard that before. That's terribly scary.
If you look at the numbers for colon cancer, you'll see they're not that great. The survival, if it comes back, and it's underserved. It's an underserved cancer.
And if it comes back, you're saying that these tumors are more chemo resistant than they were before?
Absolutely, absolutely, they're far more chemo resistant.
And your product solves that.
It solves
Because it's
it.
actually it's focused on those chemo-resistant tumors. There's something in that chemo-resistant tumor that is expressed that actually attracts your product.
It's like on the surface of the tumor, there's just more doors that open, and that door is CD147, and it's more resistance. The more doors are on the surface, and the better it works.
Fascinating. So tell us. You've now told us sort of how it works. Tell us where you are in the study approval process. Have you done any studies in humans yet?
So we want to go into a human study. We've done multiple animal studies, and we've come to a point where we're trying to raise money for, again, a phase 1 human study. And I just pointed out we want to work with patients that are terminal, which is like very unusual. I mean, I have to work with my chief scientific officer and chief medical officer, see if they can design this. They think that it's possible to do terminal fit patients for this drug instead of doing the slow way, just because we see this like remarkable results we have in mice. So again, the 25M covers malaria, which is 5M, and the rest goes to colorectal cancer.
So how many patients would you have in this $20 million study?
I didn't look at the study design. That's still being planned. But my guess is it's somewhere between like 30 and 40 patients is probably standard for phase 1.
And how long, you've already said that they would be terminal. And so, how long do they get injected once? How many times? Like, tell me about what the course of the medication is.
So I don't know the injection protocol, but you could inject it more than once. It's so many milligrams per kilogram that's administered over, so many weeks. But again, if it reflects the same efficacy as it did in the animal models, I mean, it should be highly effective against these patients. It shouldn't go on for months and months. I mean, God willing, they'll be sent home earlier and they'll be removed from their terminal state if the drug works as well as it has, if it
And if
translates.
the phase 1 works, you'd have to do the same thing, go through a phase 2 and a phase 3? We
would. There are certain exemptions you can get to allow the drug to be utilized during the clinical trial if it has exemplary results like this. Now, I'm not a regulatory expert, so I'm gonna leave that questions to maybe part 2 here with you. But
it seems
absolutely.
to me that if what you're doing is actually delivering the chemo payload more effectively, that some of the people that might be interested in this are the companies that produce the chemo payload, because they would look at your drug as a better way to deliver their drug. Is that a possibility?
Well, the payloads that we use are off patent, and they're produced by many, many companies. It's just not one. So, I think a lot of people would be interested in this drug, but it's nothing proprietary to get this payload. There are many CROs that will conjugate it to the antibody. It becomes like buying a loaf of bread at a grocery store at some point. So there's nothing proprietary to it.
So, I'm familiar with a process called a 501(b)(2) process that would take a formerly generic product through the approval process more quickly if you've changed the delivery mechanism but not changed the efficacy of the payload. Is that a potentiality of a way that you could actually get approval more quickly?
So I'm not a regulatory expert, but I would say no, because I think the regulatory agencies deem this as a totally new drug, because an antibody binding specifically to an antigen on the surface would be considered unique on its own, irrespective of the payload. So I don't think that's possible. Again, I'm not a regulatory agency, but I would say that if that's true, many other ADC companies would have done that.
Right. So you've got to start from scratch. And so the phase 1, phase 2, phase 3 would be what? The same 3 to 5 year time frame that we talked about before?
Yeah, I'd say that's correct. I'd say that's correct.
And you're going to have to, once you prove the efficacy in the phase 1, you're going to have to come back to raise more money for the phase 2 and the phase 3. But it becomes a lot easier because you got the proof of efficacy in the phase 1. Is that correct?
Exactly. I mean, if you pass a phase 1 like this and show efficacy, you'll have investment bankers knocking at your door, and large, almost every VC would want to be on that deal.
So tell me, take a step back, and you seem to be an incredibly smart guy. I took a look at your resume. You've got a great background. Why would someone like you get motivated to get invested in and committed to 2 products? They're going to take 3 to 5 years to come to the market. You got to get $25 million to prove efficacy before you even get sort of people saying you're right. Why take such a big risk?
Well, I involved myself in this business without understanding those risks. So I didn't understand the risk. My business partner didn't understand the risks, and how long it took. I mean, we've been at it for 10 years. But we're here now, so we try not to look back, and we look what we've accomplished to date, which is phenomenal. Like we could have failed right up to this point. I think we've already succeeded because few drug companies can even manage the type of efficacy that we're seeing in this period of time with the little bit of money that we did spend. So I say we've actually succeeded. But you're right. Then here's the last stage is getting the human data funds raised, and I think that's a product of the environment right now. It doesn't reflect on the performance of our biologic. It's just environment right now. You have to understand that these funds are scared that they're going to run out of cash. And they can't raise new funds because everyone's on the sideline waiting to see what's happening with all these tariffs and these adversarial trade agreements that are coming across, these are creating nervousness for investors. They want to see what happens. They want the dust to settle, especially the Health and Human Services Director in our country. They're making policies that create uncertainty. They want to know exactly what's happening before people invest. It's just human nature to pause. And so that's not a reflection of how good our drug is. It's just the environment that we're in.
It seems to some people internationally that Robert K. Jr. is almost questioning new technologies, new products, new ideas, and so that there's a cloud over, clearly new vaccines, but like new science. Is that part of your problem?
Well, so far we don't have an mRNA product, even though our malaria drug could be an mRNA. It could be. That may not be affecting us directly. I mean, there's also a flip side. There's also something good to say about his policies. I mean, he recently announced that they are trying to move away from animal testing, monkey testing, primate testing. And in that respect, it could be helpful to us because we can test these in mice. We don't have to do primates. So sometimes it's a double-edged sword. It depends on the industry you're in, and it also depends on what you want to have achieved. But you have to be very careful and look at each thing independently.
The big challenge. We're going to take a break for some messages, and we're going to come back with PJ and Murat and chat a little bit about how you actually raise money. We're going to go from the science to actually raising money. Stay with us, everyone. Back in 2 minutes. Welcome back, everyone, to the Brian Crombie Radio Hour. I've got Murat Croci with us today. He's the co-founder, the co-CEO of Ibex Bio. They've got two fascinating products that have gone through animal testing. He's ready and poised to take them into humans in phase 1 studies. He's out trying to raise $25 million, $5 million for a study on malaria and $20 million for a study on refractory colon cancer. And it sounds very promising. And he's talking about the challenges in the marketplace. We've also got his investment banker, Pierre-Jean Fournier of Fournier Investment Bank out of Paris, France, who's an expert in raising money for biotech companies, etc., and has been involved in this business both in Europe as well as North America for 2 decades now, I guess, PJ, if not more, frankly. Let's talk a little bit about the process. So you're a private company, Murat, I presume?
I am, yes.
And so you've got to go to private investors and raise money. I presume you've got a website that's got all of your science and a investor deck on it. But people are going to be making not an investment in a public company through a public security. So what is it they're doing? They're taking a look at what your investor deck and is there an information memorandum there or a prospectus or something like that?
So it's a 506c offering. And basically, if you're an accredited investor, you can look at the investment agreement and all the information and make an investment right on the website.
An accredited investor is what? Someone that makes a certain amount of income or a certain net worth and so therefore is deemed to be sophisticated?
That's correct. That's
okay.
correct.
And you have to do a compliance type application to prove that you're an accredited investor before you get access to the information?
So if they don't have a lawyer letter, if they don't have an accountant letter, we would help pay for that certification. So they would just click on the website, please help me certify. And we would send it to a third party to help them get certified. It's just very
So then once they get certified as an accredited investor, they can get access to the information and they go through it. And then what you're asking for 25 million, but I'm not one person putting in 25 million. How much have I got the right to put in?
5K. That's the minimum investment.
5K is the minimum investment. Okay. And of the total $25 million, does that buy 100% of the company or 50% of the company?
So the valuation, so it's 2 parts. The offering is 37.5 million is a pre-money valuation, 37. So 50% goes into the 37.5 million. The other 50% is a convertible note which is at 12%, and it gives the investor the option to convert at a 20% discount their principal and interest. So it's
So that's interesting. So $12.5 million goes into a convertible note. Take a step back and explain the convertible note, why that makes sense.
Okay, so if someone invests 10K as an example, 5K goes into equity at 37.5 million. The other 5K is a convertible note. It's again 12% with a 20% discount. It makes sense for a couple reasons. If the valuation's off, the investor can make it up. They get also precedence over equity positions because it is a kind of form of debt. So again, if the valuation doesn't go higher, then they can make up the difference with the convertible note and the discount.
Oh, so it's a 20% discount to the next offering. Is that correct?
Correct. That's right.
So that's interesting. So you've got downside protection with the convertible note, and some upside exposure with the equity, and in a complete sort of failure, you're in a debt position, so you've got some security in the debt position. If it's a down round where the next round has a lower price, you've got the protection of you can convert at a 20% discount to the next round.
Absolutely.
And then if things look spectacular, you can convert. Is the conversion right at my choice, the investor's choice, or can you force me to convert?
It's at the investor's choice, and it's at the next round's valuation, whenever that is.
And how long does the debt? How long is the debt outstanding, and how long is the conversion right there?
It's within a 4-year period.
Sounds fascinating. And then, what would be your intention? You would go out obviously after phase one and do another private round, or would you be going public at some point in time in the future?
We'd love to go public if the public markets open up. We'd love to list this on a public exchange and get an exit for our investors in that manner.
PJ, what do you think the chances are of Murat being successful in this?
Well, I'm going to move the result to 100%. You asked me about how is it going to raise funds in the case of Ibex. You have to understand that the ADC market is very, very recent. So much so that I signed 5 deals in this sector over the last 3 months. So it's a very recent market, very effective against cancer. And again, for the public, the mechanism of action is very simple. It kills the cancer cells selectively, okay, targeting the cells. So I'm very, very optimistic.
So you don't lose your hair?
No, I didn't lose my hair because I put a hat with ice on it, so it prevents the hair to fall down.
No, but with this drug that Murat's got, you don't lose your hair because it's selective and it doesn't kill hair cells?
Makes you laugh, Murat, so I don't know.
Well, it sounds like it's fascinating. Murat, is there a website people should go to if they're interested?
So ibex.bio. ibex.bio. They can click the invest button and they can get more information from there, but they can see the website at ibex.bio.
And if you want any information from me, just contact me at briancrombie.com, and I'd be happy to forward your interest to Murat and to PJ. PJ, before I let you go, I understand you've just published a new book. What's the new book? This is fascinating.
The title is How I Beat Goldman Sachs.
How I Beat Goldman Sachs. You beat Goldman Sachs?
Yeah, it happened 5 times in the late 90s, and one of them, I got them the big deal of the year. It was the PMC, Philip Morris Corp, which was the biggest company on earth at that stage. And I took the deal, which was $10 billion underwriting against Goldman and DLJ and Citibank. And that was amazing because I was on my desk dealing with 500 investors in the world. A lot of fun. You should check the book. It's very funny to read.
How I Beat Goldman, and how do I get it? Through Amazon or what?
Amazon. You type my name.
Excellent. PJ, thank you so much for joining us. I really appreciate it. And thank you for the introduction to a fascinating gentleman and a really innovative company, Ibex, that I'm hopeful gets the money, because I think this is going to make some important moves forward in life and health. And so Murat Croci, thank you so much for joining us and telling us a little bit about your product for malaria that is right now killing 600,000 people a year, and this product sounds like it could really solve that. And also your product for refractory colon cancer that takes the chemo payload and delivers it far more effectively, particularly to people that have got colon cancer that come back 5 years later with the cancer all over again and really need your product. So thank you so much. Ibex.com is the website to go to, or Brian
Not
at
bio.
briancrombie.com.
Not bio.
Sorry, ibex.bio. I apologize. Thanks for correcting me. That's our show for tonight. I'm going to come back with a couple of concluding comments in just a minute. Thanks very much, everybody.